Hello everyone, welcome to another video. And since we were covering neuroradiology in the last video, so I will be continuing with that topic. And today, what I am planning to discuss is congenital supratentorial brain anomalies, and it's a very lengthy topic and concept. So we will be dividing this in few parts. So this will be the first part, and we will be discussing about key malformations, their embryological origins, and what radiologists need to know. How to understand brain malformations. This is basically we cannot learn these conditions as in like individually the brain malformations, but we need to build a framework. So brain malformations are best understood by when the insult occurs during development. So to understand that, we need to have a core idea about during embryogenesis, what is happening when. So in 3 to 4 weeks, neurulation occurs, and if there is disruption at that time, there is insult at that time, then we end up with neural tube defects. And later on, from 8 to 16 weeks, neuronal proliferation is occurring. So if there is any issue at that stage, then the issue arises like microcephaly or megalencephaly, or uh, things related to like small brain or large brain, to be more crude. And then coming up to 11 to 15 weeks, neurons start to migrate upward or outward. So if there is any insult at that time, that disrupts neuronal migration, and that is when we have Lissencephalies or heterotopias that we will be discussing today. And lastly, from after 15 weeks, there is late migration or post migration, and during that time, cortical organization is occurring, and that disruption at that time will produce polymicrogyria, Lissencephaly, and focal cortical dysplasias. So the key thing that we need to understand is the timing of the insult determines the malformation. So another table and another timeline to know the normal brain development. So there is neurons are born in germinal matrix and from form neuroblasts, and then they migrate radially outward, and then there is organization, and then they form normal cell gyri. Interrupt this process at any stage and you get a predictable malformation. So another division, like if there is failed migration, there will be heterotopia. If there is incomplete migration, there will be Lissencephaly. If there is abnormal cortical organization, there will be polymicrogyria. And if there is abnormal cleft formation, like post-migration thing, there will be Lissencephaly. And now this is another diagram explaining the neuronal formation and migration. So in the last uh, line, you can see that there is microcephaly, then there could be hemimegalencephaly, polymicrogyria can occur at this stage, then we have focal cortical dysplasias. These things are after post migration, and then we have Lissencephaly, periventricular heterotopias, which are during migration things. And agenesis corpus callosum is also part of the spectrum that we will be discussing in detail in the next video. So now we come up to the simplified version of cortical malformations. Here is a quick reference table. 6 disorders, 3 processes. I have highlighted the 3 similar ones in pink, which are Lissencephaly, pachygyria, and heterotopia. These are basically problems with migration. If there is very poor migration, there is smooth brain. Lissencephaly means smooth brain. Pachygyria means incomplete migration, like there is some migration, and heterotopia means arrested migration. And gray matter then is arrested in wrong place. And after, if there is abnormal organization after the post migration stuff, that would be polymicrogyria. And if there is some cleft formation, that would be Lissencephaly. And if there is focal cortical dysplasia, that would indicate blurred gray white junction. So we will be discussing all of these. Now coming up to Lissencephaly spectrum. Lissencephaly is not a single disease, it's actually a spectrum from most severe to mildest. Agyria means complete absence of gyri, A mean absent, smooth brain. Pachygyria means broad and thick gyri and reduced sulci. And lastly, we have subcortical band heterotopia, partial migration of the neurons leading to double cortex. And these are not separate diseases, these are actually part of the same neuronal migration spectrum. So we need to know that this is severer to milder forms, and they are all basically have the same core concept, the abnormality in migration. Now let's discuss some types, and let's have a review of this image. On this MRI, you can see that there is marked diffuse thickening. Let me on the pointer. This is diffuse thickening of the cortex, and this is more than normally cortex is 4 to 6 mm, but this is more than that. This is more than 1 centimeter, and the outer surface of the brain is very smooth. Like there are very few poorly formed gyri and smooth bilateral cerebral hemispheres, giving the classic figure of eight or hourglass appearance. And there is also dilated ventricular system, and this is classic appearance for Lissencephaly 1. We have written that in here as well, whatever we were seeing. But let's review what is Lissencephaly 1. So we already know that this is an issue with migration. So basically there is under migration and neurons fail to reach the cortex. So neurons are not reaching the cortex, and there is not formation of sulci and gyri. So this is smooth brain, thick cortex, shallow sylvian fissures, figure of eight or hourglass appearance on axial imaging, and posterior cortex is more involved than anterior cortex. These things are important for MCQ points and sometimes distinguishing while reporting as well, so this is important. Now just for this genetic table is important because sometimes these can appear in SBAs. So if this gene LIS1 is involved, then the pattern basically would be more about posterior predominant disease. If DCX disease gene is abnormal, then there is anterior predominant Lissencephaly, and T2BA1A gene is involved, then there is severe brainstem and cerebellar anomaly. Now we have another thing which is Miller-Dieker syndrome. This is the most severe and recognizable form of type 1 Lissencephaly on prenatal imaging. The on MRI there is grossly abnormal brain outline, few shallow sulci, shallow sylvian fissures, figure of eight appearance, and cortex is markedly thickened, like around 2 centimeters. Craniofacial abnormalities will be there, like microcephaly, bitemporal pitting, prominent occiput, micrognathia, flared nostrils, and there will be association with congenital cardiac issues, renal issues, as well as cryptorchidism. Prognosis is very poor, mostly do not survive beyond infancy, and there is also 25% risk in future pregnancies. So this is important, especially for MCQs, and during day-to-day practice we can and we should know about this while especially doing our fetal imaging. Okay, so now we have another CT scan, and this CT scan is showing us that there is less lobulations along the cortex, and there is some irregular cobblestone like appearance of the outer cortex. We can see a VP shunt on the right side, and there is not much ventriculomegaly, and the hourglass appearance is still there. So what is this? This is basically type 2 Lissencephaly, where we saw right-sided VP shunt, smooth cerebral surface with thick cortex, diffuse white matter hypodensity in both cerebral hemispheres, which basically reflects hypomyelination, which is a hallmark of type 2 cobblestone Lissencephaly. Let's review this, and you can appreciate that the white matter is quite hypo, and that indicates hypomyelination. And then we have no evidence of ventricular dilatation and sparse cortical sulci, and mild left frontotemporal subdural collection was there, which was maximum thickness up to 8 mm. So this basically is representing Lissencephaly type 2, because the main differentiation feature is that there is smooth surface of the cerebrum with thick cortex, sparse cortical sulci, and white matter hypodensity. So these were the three things that indicate that this is type 2 Lissencephaly. And for type 2 Lissencephaly, these are the key features that we need to know, that this is basically due to overmigration, cobblestone cortical surface, lack of normal sensation, Sylvian fissure will be still small, hourglass appearance is there, hypomyelination is there, ventriculomegaly can be seen but less common than type 1, and brainstem and cerebellar anomalies can occur. Now, so what is more important for cobblestone Lissencephaly is that it is associated with at least three syndromes which are to be known, and these are part of congenital muscular dystrophy syndrome. The first one is Walker-Warburg syndrome. This is very severe, and eye and muscles both are involved, and there will be cobblestone Lissencephaly with this, as well as cerebellar and brainstem anomalies. Now the next one is Fukuyama congenital muscular dystrophy. In this, eye is not involved, but there is muscular dystrophy, cobblestone Lissencephaly, and cerebellar cysts. Third one is muscle eye brain disease, that will be about muscular dystrophy, eye anomalies, as well as cobblestone Lissencephaly. So this is important, and particularly in muscle eye brain, eye anomalies are glaucoma and myopia. So these are the terms that we should be aware of, especially they can appear in MCQs. And next up, we have differentiation, the table to highlight the differences between type 1 and type 2. As we have already seen, that type 1 has a smooth cortical surface, type 2 has irregular cobblestone like appearance. Cortex is thick in both. There is failed migration in type 1 and over migration in type 2. Sulci are almost absent in type 1, however, these are irregular in type 2. Then there are some associated syndromes and difference in ventriculomegaly, and posterior fossa is more involved in type 1, and eye anomalies are more common in type 2. So, this is very important and high yield to know. Now, what coming up next, we have difference between Lissencephaly and pachygyria. So in Lissencephaly, basically the sulci and gyri are absent, but in pachygyria, sulci are reduced and gyri is broad or thick. Severity, as we can understand that when cells and gyri are absent, that's fair, and if they are reduced, then that's a milder disease in pachygyria, and cortex will be thick in both of these. This is all since these both entities are part of the same spectrum, so we can just know the difference in severity. Coming up next, we have another entity which is basically heterotopia. So, what is heterotopia? Heterotopia means gray matter in the wrong place, and it is caused by arrested neuronal migration. The migration started or sometimes didn't even start. So, how it happened, let's see. There are three types. This could be subependymal, this could be subcortical, and there could be band heterotopia. Subependymal means nodules along the ventricles, subcortical means gray matter within the white matter, and band is the double cortex. And the universal rule that we should know to understand and diagnose heterotopia is that it follows the same signal as cortex on all sequences. So let's have a look at one image. Sorry. Okay, so here we can see that the ventricular surface appears irregular, symmetrical bilateral, And this is a contrast study. We cannot see any contrast enhancement in the nodules seen along the subependymal region. And these are following the signals of gray matter, i.e., cortex. So, what is this? This is basically subependymal heterotopia. And what we are seeing is that gray matter nodules are lining the walls of the lateral ventricles. Neurons are arrested at the germinal matrix and they couldn't migrate outward. This is the most common form, and this is mostly exclusively in females. And if it happens in males, they mostly die in utero. And it presents with epilepsy and near normal or normal intelligence, and seizures typically begin in second decade. So the MRI features that we need to understand are the nodules along lateral ventricular wall, subependymal, do not suppress on flare, no enhancement, follow the gray matter, no edema or mass effect, and they could be unilateral, bilateral, single, or multiple. And how to differentiate these nodules from tuberous sclerosis? Those nodules can calcify and may enhance, but heterotopia will never enhance or calcify. So this is a must-know difference between these two nodules. Now coming up to another image. In this image, we can see that there are some gray matter areas that are visible here, like in these areas. So these are some abnormal gray matter areas in the posterior cortex. So what could these be? So let's understand that we are reviewing an MRI in which there are nodular heterotropic gray matter within the white matter of the posterior spectrum of the right temporal as well as occipital lobes, and they appear continuous with the cortex and indent the occipital horn as well. So this is basically subcortical heterotopia. So, what is subcortical heterotopia? In this, nodules or masses of gray matter are rested within the deep white matter, deeper than the band heterotopia but not touching the ventricular wall. This is rarer than subependymal, sporadic mostly, can be unilateral or bilateral. It was unilateral in our case. And epilepsy is this presenting feature, and overlying cortex may appear thin or dysplastic. So these could be nodules or curvilinear masses within the white matter, not touching the ventricles. Isoin tends to gray matter, no enhancement, no flare suppression. Overlying cortex may be thin or malformed, and white matter volume can be reduced on the affected site. The deeper and larger the heterotopic mass will be, the more severe the clinical presentation will be. Size correlates with disability. So, this is important to know that symptoms can guide you to the severity of the disease in heterotopias. Now, we have another image. This is pretty simple. In this image, we can see that we can see normal cortex, near normal cortex, but beneath that there is a similar signal band which is seen below the normal cortex, and this is visible here. This one, a whole band is seen along almost whole cortical surface, below the whole cortical surface. So what is it? This is known as double cortex. So this is bilateral symmetrical thick band of gray matter, deep to and running parallel to the cortex, simulating the double cortex sign. Overlying cortex exhibits mild shallowness, however, agyria or pachygyria is not visible. So this is called subcortical heterotopia. What is subcortical heterotopia? This is also called double cortex. Neurons partially migrate and arrest in the subcortical white matter, forming a band of gray matter beneath the cortex. So some migrated, some stayed back. And this is basically thought of as being in the Lissencephaly type 1 subcortical band heterotopia spectrum. So this as the genetic defects are basically same in both the diseases, Lissencephaly 1 and subcortical band heterotropy. This is not to be confused with the subcortical heterotropy that we saw earlier. So here the neurons do migrate partially and arrest in subcortical white matter, and if it is more common in females, and if there is DCX mutation, then there will be classic Lissencephaly. So same genes but some spectrum of the presentation. Both are two ends of the spectrum, Lissencephaly and subcortical band heterotopia. Overlying cortex can appear normal. There will be band of gray matter beneath the cortex. White matter is trapped between the band and cortex. Signal will be identical to the cortex, and anterior posterior gradient varies by gene. DCX anterior and LIS1 posterior. And bands may be complete, incomplete, or duplicated. Two concentric bands can be seen, especially in the temporal lobes. So this was all about double cortex. Now let's see our table to revise these heterotopias with differences. We can see that all but we just reviewed that subependymal is within ventricular wall, subcortical is in the deep white matter, and band is between ventricle and cortex. And appearances vary, and then we have genetics and signals will always be following the gray matter. They will never enhance, and seizure onset is variable, and the part of spectrum is only this one band, the double cortex one. Now let's have a look at some SBAs. You can and you should try to solve these. This is the first one. Now let's review the second one. The third one. This is our fourth. And here is the last one. So these are almost from the material that we just discussed, and lastly these are the answers to these SBAs. So review these, and if still there is some confusion, try to revise the video. This was all from my side for part 1 of supratentorial congenital brain anomalies. In the part 2 we will be discussing a lot in the same terms, in the same category, and I will be keep making these SPAs as well. So that's all for this. These are the references for today's video. So subscribe and stay tuned for the upcoming videos. Bye bye.